Hi, folks. My name is Jeff Johnson. I'm CEO of Premier Bee in Sioux Falls, South Dakota. And today it gives me great pleasure to be able to introduce to you a video that we have Dr. Marla Spivak speaking on Propolis envelope. And the Propolis envelope really is what led to us developing the Propolis hive. We introduced the hive at the North American Honeybee Expo, which is also where Dr. Spivak was one of the keynote speakers. And what you're going to see today is a video that takes some outtakes of her lecture at the North American Honeybee Expo. Enjoy it. It's really good. Dr. Spivak has spent a good portion of her life figuring out why Mother Nature wants honeybees to have a Propolis envelope in their home. Yeah, enjoy the video. Thanks.
How many of you hate this stuff? Go ahead, you can admit it. But I'm here to convince you that it's really actually good for bees and that you should start paying attention to it a little bit. So what is it? Propolis is really a plant resin. Some plants secrete resins. It's a plant defense. So these plants that make resins, secrete resins, do it to protect themselves. And they protect from infection and from UV light and from herbivory. Thank you very much. I appreciate it. So the plants are producing these highly antimicrobial compounds to protect themselves, and honeybees are amazing, as you know, and they can locate these plants that are producing or secreting resin, and they go and they scrape it off the tree. In the left-hand photo, you see a green plant, and that's actually in Brazil. It's in the genus Baccharis. And it's a shrubby plant, and that bee is scraping a green propolis right off the leaves. They scrape it off the leaf, they handle it with their mouth parts, and then they transfer it with their legs, just like they do pollen, and stick it on their back legs. And then they come back to the nest, and it's so sticky, they really can't get it off of their back legs by themselves. So another bee in that third photo is kind of helping remove the resin from the back leg. And then together, the forager, the resin forager, and the bee help cement it into the interior of the hive cavity. So they're not eating this stuff. They're not normally, especially here in the US, getting the resin from plants that are producing pollen and nectar. Some bees are doing completely different foraging to totally different plants just for these resins. And once they bring them in, as far as we know, they may be putting some salivary secretions in there, but they're really not changing the chemistry of the resin. They add a little beeswax to it, depending on the gaps that they're filling, but they're not really modifying the resin. And they put these resins in a wild tree cavity. They're lining the whole interior with a layer of propolis. Well, inside the hive, we call it propolis, and I'll probably use resin and propolis interchangeably, those two words. I think we knew about propolis long ago for the medicinal properties for humans. And so they thought the bees were producing the propolis. And they didn't know that it came from plant resin. So maybe if they had known that, they would have called it resin inside the hive, but we call it propolis or propolis. It doesn't matter how you pronounce it. But they use it to line the whole interior of the nest cavity in a bee tree like is shown here. The antimicrobial properties of propolis provide everyday benefits to bees' immune system. They really help their immune system be stabilized and function well, and it helps stabilize and promote their core microbiome species. It doesn't act against it. It actually helps their microbiome, and it helps reduce disease load within the colony. And tomorrow I'll show you the data on those. But right now, like I said, I'm going to just talk about how to encourage bees to make a propolis envelope. I mean, we can keep them in trees, but then it makes it kind of difficult to manage, right? So how do we help them do that? This photo is a bee tree that we got at the university, and you're looking as if you were a bee, you're looking out the front door, because bees tend to put a lot of propolis, especially around the entrance of a feral colony, of a tree like that. And so all of that orange and yellow and brownish stuff, that's pretty thick layer, maybe up to a 1/4 inch layer of propolis right by the entrance, but they do put it all around. And they don't put it in your Langstroth boxes. They do put it in the cracks and crevices and everywhere where it annoys you, under the frame rests, between the boxes, making you use a hive tool. You probably hate it for that reason. And beekeepers in the US have not liked bees that collect a lot of propolis. It's a genetic trait to bring in a lot of propolis. They don't like it because it makes managing the colonies very difficult. But if you look inside the boxes, because they're plain smooth, the bees are not attaching propolis to those smooth walls. In contrast to the photos below, in a tree cavity, they just like varnish or paint the walls with propolis, and like I said, especially around the entrance. So what's going on here? How do we, if we're working bees in Langstroth boxes, how do we encourage them to make that envelope? And some bees, colonies collect a lot of propolis naturally. It's, like I said, it's genetic trait. Some of them really fill up cracks. You've probably seen this, where the propolis in warm weather is kind of oozing out of the cracks in some colonies, on the tops of some frames, especially in fall, late summer, they tend to close up the entrances or reduce the size of the entrances with a lot of propolis, some colonies like that. Those are colonies you might think about raising some queens from. Okay, so to make this propolis envelope, what Mike did first was just collect propolis from different areas. You know, you can just scrape it off the sides of boxes and collect it, and then make a tincture or an extract by dissolving it in 70% ethanol and making a 30% solution, okay? This is, I don't recommend you do this. This is what they do if you're selling propolis for human medicine. They have different extracts they'll use, different percent alcohol or a water extraction. But we just kind of made a simple dissolving the propolis in ethanol in a dark container and shaking it for about a week and then straining it, and then you can figure out your percent solution. And we, that's the royal we, it's all Mike, okay, painted it inside these. We just started with small colonies with 5-frame nuke boxes, and we painted a known amount, same quantity of propolis around the boxes. And then we had controls where we just painted the ethanol in case it had some activity also. And another way that we tried, that Renata tried when she was a student, is we took propolis traps, which they sell. I didn't see them. I haven't gotten around the whole expo yet. But there are these propolis traps. Have you seen these? You know, they come in different colors. This one's black. And they have about a 1/4 inch gap in them, 7 millimeter gap. And for some odd reason, bees like to fill the 7 millimeter gap with resin or propolis. And if it's a little bit wider, they start adding a lot of wax in there. So this particular gap is this magic thing for bees where they only put resin in it. And if you put it on top of the colony, and in this case, you can see the photo, we put a rim around it, we let some air in, especially if there's air and sunlight. Bees don't like that on the top of their colony, and they really fill those gaps up with propolis. So what we did was take those propolis traps and cut them and staple them inside of bee boxes. I don't recommend you do this either because it really messes up your bee space, especially when you put them in front to back, okay? But this is what we did for our experiments. And yes, the bees filled them up with propolis, you can see. And if you remove the trap, this is what the, quote, propolis envelope looked like, the photo on your right. We left them on for the duration of our experiments, so the bees would just continue to add on. So that was the second way that we made some propolis traps, okay? And for our research, we were able to see in both methods, with Mike's painting on the extract and with this method, we were able to see all the effects on the immune system and the diseases that I talked about earlier. So those were effective, but they weren't great for beekeepers. So then we started playing around with different kinds of mesh. And this green kind of a soft mesh, like those bags, like an onion bag, but a finer mesh. And those work, except the bees shred them up and then you start seeing them in the honey supers. Don't do that. Okay, this is not a good method. We tried some harder screens, plastic screens. These are just shown on top of the colony, but we were able to, put them inside the inside walls. And those work. They're kind of variable. They're not very thick, deep. So there was just like a layer of propolis. But mostly we were wanting something more natural. We didn't really want to put more plastic in a bee colony. And when we went back and thought about the trees, they're just super rough interior. They're rough and they're craggy and they have deep gaps like that. And that's where the bees are really putting the propolis in. So we thought, how can we do that to our colonies? So we took a wire brush, scraped them up, and like that, you can just see. And yeah, the bees kind of put a sheen, a little bit of propolis there, but not nearly like they do in a tree. Plus, it was a lot of work. And I didn't want to do it. And then a commercial supplier made some boxes with these smooth ridges, okay? Now, they're ridged, but they're smooth. Okay, and I don't know the tool that they used to do this, but when we put them in the colonies, yes, they put in some propolis, but not anything like they did in a tree cavity or like they did in those propolis traps. So then we met this guy who lives in northern Minnesota, Christian Dom, and he's not selling these anymore. He fells his own trees and he makes his own lumber, and he worked in the lab for a little bit, and he made some boxes with these 1/4-inch grooves that were very, very rough, and gave them to us to try. And they were amazing, okay? So the bees readily filled those grooves with propolis, okay? And the point is that the smoothness wasn't it. It's got to be really rough, and they've got to have these, at least 1/4-inch gaps, and as deep as you can get without compromising the integrity of the wood. So after one season, some colonies, those that, have this genetic trait, really bring in a lot of resin. And the resins come in different colors, these propolis. This one on the left is kind of reddish, and on the right kind of gray from different areas, different trees, sources probably. So one study that I will talk about is we used those rough boxes in a big study. Mike used those rough boxes, and we collaborated with one of my more current students, Maggie Shanahan, and the ADs from 80 Honey Farms, who allowed us to use some of their colonies. So we gave them some of these rough boxes, and there's the publication that is still in press somehow. It is accepted for publication, and it should be coming out soon. The reference is at the bottom there. So we started, Maggie's part of it in Minnesota was she compared a smooth box with a box with the propolis traps in them, because we'd had a lot of data on that, the benefits of having that kind of trap in there, and then some of Christian's rough grooved boxes. And we hived colonies in these and compared them. And here's kind of examples. The smooth wood, they do bring in propolis. Like I said, it's in the cracks and crevices and sometimes around the entrances. but not nearly as much as with the Propolis draft or with the grooved boxes. And in the commercial operation, we looked at 120 colonies over 2 years, and 60 of these colonies were hived in boxes with these that are roughed and grooved like this, and 60 were just standard Langstroth boxes. And then Mike and others followed them around, tagged those colonies and followed them on their commercial migration. So they came from Mississippi, they went up to South Dakota for honey, they went over to almonds in California in February. So they looked at the colonies in August at the honey harvest in year 1, and then they sampled them in almonds in years 1 and 2. And this was right during COVID, so we were kind of limited on how much, especially from the USDA, giving permission to workers to go out and work was difficult. But he persisted and took all kinds of measurements. So how much propolis they deposited, how big were the colonies in fall in South Dakota, and then during almond pollination, how much honey they produced. If they were, the question is, if they're bringing in a lot of propolis, does that compromise honey production? And then all kinds of measurements of their health. Do they have signs of disease in them? What's their immune gene expression, which I'll talk about tomorrow, and how does their microbiome look? Okay, so some data. You're okay with data, right? Oh yeah, okay, good. So the controls, those with no standard boxes, are in orange, and the yellow one is from Maggie's experiment in Minnesota, where she put in those propolis traps, and the blue is the rough boxes in these cases. So the stationary data is just the data from colonies that stayed in one place for one season in Minnesota, and from April through August, you can see the colonies with the rough boxes produce the most propolis. And the way we did this was to take the boxes away from the bees for a little bit and photograph the walls. And then you use JPEG, right, Mike? Yeah. To quantify the density or the pixels in the photo to figure out how much propolis was in there. So it's an approximate measure, but it works pretty well. And so you can see the rough boxes, the bees put up a lot more propolis. And in the migratory colonies, by the end of the first season, by August, there was a little bit more. The B shows that's different from the A, that shows they're statistically different from each other. And then those same boxes, by February, the rough boxes had a lot more propolis. And by year 2, in February, same boxes, a lot more propolis. So interestingly, it took them almost 2 years to make as much propolis in the migratory route as they did in Minnesota in just a few months. Okay, this depends on the trees they're getting it from in this case. Because of all those 60 colonies or 120, some of them have to have that genetic trait. Okay, so this depends on the landscape that they're in. And the diagram on the right shows the landscape that they were in. So the left-hand bar where it's all the colors in the stack show all the land use in 2.5 kilometers, so about 1.5 miles surrounding the colonies. In Minnesota, you can see there was a lot of different land uses. So there's forest and flowers and agricultural land, and the blue is corn and soybeans. And you can see in the migratory one, in different areas, there's just not as many of the flowers, the red and the orange and the yellow, the wetlands that might provide or have the trees or sources of resin for the bees, okay? So that's probably the difference for how much they collected. And what plants did bees collect resin from? We have no idea, but from our research in Minnesota, like as I said, they are collecting most of it from cottonwood. Isn't that funny? I would not have ever expected it. But now when I go by a cottonwood tree and you break off a branch and you smell it, you're gonna go, oh, it smells like my colony. We also saw, I love to say we, I like to take credit for Mike's work. These graphs, even though they go upside down, you just look at the difference between them. So let's look at the right-hand graph first. That's the signs of European foul brood in the colony. So that's actually counting cells with European foul brood and then standardizing it by how much brood they had in the colony from our estimates of brood area. And they had lower amounts of EFB if they were in rough boxes. It's not statistically different, so we can't say it was different from a research point of view, but it looks really enticing. And when we collected bees from those colonies and looked at their gene expression, how much EFB was in their bodies, these would be in the adult bee bodies that are carrying the disease, the bacteria, there was significantly less, the blue bar is lower than the orange, significantly less EFB gene expression in the bodies of the adult bees. So they weren't carrying as much. And I think Mike will be, and hopefully others will be following up on this research coming up, because this is really enticing. Because EFB, I don't know, there just seems to be more and more of it, and tricky to treat sometimes. Are you seeing more EFB? All right, some of you are. So that was a new finding. For Varroa, you can see on the left that, again, the rough colonies had a little bit fewer mites in there. These are mites per 100 bees, and they were way low. They were all under 1 mite per 100 bee anyway, because the commercial guy was really controlling mites well. So the difference between 0.6 and 0.4 mites per 100 bees is hardly anything at all. There is a trend. You can see the blue dots versus the orange, that colonies with the rough boxes tended to have fewer mites. And that's a good thing. Any help is good, right? Even if it doesn't treat them, it's a good help. These are complicated, but these are measures of the immune system, and I'll talk more tomorrow about how we measure this. But in general, what this is saying is the propolis is helping stabilize the immune system. So your immune system is, in humans and in bees, is really a costly system to run. If your immune system's always ramped up, that's hard on your body on some other function that you might have. So keeping the immune system at a stable hum is a really good thing, okay? Increasing, you can see there were few antimicrobial peptides. These are just proteins in the immune system. There were few that actually increased the immune gene expression, and one that was destabilized, but mostly the immune system was decreased or stabilized. And by decreased, it just means that the propolis, because it's so antimicrobial, it's doing the job for the immune system. It's killing off microbes in the nest cavity so the bee's immune system doesn't have to do it. That's all I have for this particular talk. In conclusion, the use of rough interior hive boxes is good for bees. It will help their immune systems. It will lower their disease load. It will not compromise their populations. If they're a good, strong colony, it will not compromise honey production. And there's no reason to not be using rough boxes. And we're proud to announce, although I'm not allowed to endorse or promote any company, there is a company out there that's now selling these rough interior boxes. So I hope I'm not selling, don't get me in trouble, but I hope you try them out on your bees and then let us know, or let Mike know how they are.